to introduce my research work. The work I'm going to share with you today is about identification of chilling-tolerant genes via backed segregant analysis sequencing. As we all know, the region of maize is Mexico. After Colombia discovered the map, he brought maize to Spain. With the development of the world's navigation industry, maize gradually spread to all over the world and became one of the most important food crops. At present in China, maize is widely planted in various regions, among which the spring maize planting area in northeast and North China is the largest maize planting area. However, in the northeast region, low temperature cold damage occurs almost once every 2 years during the sowing season, causing large areas of spring sow maize to freeze to death or get injured, resulting in a significant reduction in maize production and seriously threatening food security. Therefore, conducting genetic improvement of maize cold tolerance and enhancing its tolerance to low temperature is of great significance for ensuring high and stable yields of maize. In our experiment, it was found that different maize environments should vary large differences in low temperature tolerance. We planted different environments in the same pod and found that before the low temperature treatment, there were no obvious phenotypic differences in their growth and development. But after low temperature treatment, some environments would survive completely, some would die completely, and some would survive partially. We conducted a cold-tolerant phenotypic observation population consisting of 331 environments. We conducted a cold-tolerant phenotypic investigation on each environment in this population, using the survival rate as an indicator. It was found that the survival rates of this population after low temperature treatment ranged from 0 to 100%. The cold-tolerant trait belongs to a complex competitive trait. In our research work, we used a relatively novel research method, that is BSA, backed segregant analysis combined with sequencing, to identify quantitative trait loci related to maize cold tolerance. BSA combined with next generation sequencing was initially referred to as QTL-seq. This method was first reported in 2013. They took rice planted as a target trait. Target was A and B.
Sorry for interruption, doctor. Actually, you are still in the first slide. Could you please move your slides?
Sorry. Can you hear clearly?
Yes, you are still in the first slide, doctor. Could
Okay,
you please move your slides in the Zoom where you have shared your screen? I think you are sharing your screen in separate PPT. Please share your slides or screen. Please move your slides in the Zoom. Just move to the second slide once.
Can you hear me?
Yes, yes. Please move your slides.
Oh, sorry. I can't remove the noise.
Okay.
Can I continue?
No, doctor. Actually, you are still in the first slide. It is a title page. We are seeing only your title page.
Oh, but I can't remove noise now. Sorry.
Move your slides. Just move to second slide once.
Um,
Click on the second slide. Could you please stop your screen share and start it again?
I'm sorry. I'm sorry. Can you hear me clearly now?
Yes, we can hear you, doctor, but we are able to see your only first slide.
Um,
Please move
I
slides to second slide.
but I cannot.
Please.
I cannot improve it now. So I continue now. Um,
Could you please stop your screen share and start it again?
I try again, but the situation is not improved now. Yes. Can you see the screen now?
Yes, doctor, we can see your screen, but it is still in the first slide. We can only see the first slide. Start from beginning.
But the screen.
Please start from the beginning. Please start the presentation from the beginning.
Okay, let me try again.
Yeah. Yes, doctor, we can see the fifth slide now. Please continue your presentation. Move to sixth slide once.
Can you see the screen now?
Yes, doctor. Now it is okay. Move the another slide. Just move
Okay.
the slide.
Okay. I'm sorry. They took rice planted as a type tree. Animals A and B have low and high stature. They were close to produce F2 population. The individual planted phenotypes in the F2 population were segregated. They sampled DNA from 10 to 20 individuals from each extremity in the bottom to generate high spot and low spot. Each of the bug DNA was applied to a whole genome resequencing. They evaluated the proportion of substrates corresponding to each of two printed genomes that can be discriminated by single nucleotide polymorphism available with two. After aligning the sequence data to the reference sequence of either of the two parents, they counted the number K of substrates having SNPs that are different from the reference sequence. The proportion of K in total shortwaves having a particular genomic position was defined as the S&P index. For example, the genome of capital A was used as a reference genome. If the genomic sequence of capital A is C at this position, then mountain have two individuals. 6 individuals shown in red have the same sequence as capital A, which is C. Then 4 individuals shown in green are different from capital A at this position, but the same as capital B. Then this S&P index is recorded as 4 divided by 10 equals 0.4. For another example, if the sequence of capital A at this position is C, but all 10 individuals have G at this position, which is the same as the sequence of capital B, then this S&P index is equal to 10 divided by 10 equals 1. So an S&P index equal to 1 represents that the sequences of the actual individuals at this position are completely different from the reference sequence, but completely the same as other parental sequence. An S&P index of 0.5 means an equal contribution of both parents' genomes to the backed family. Accordingly, the S&P index is calculated for all S&Ps detected between two parents, and the relationships between S&P index and S&P position in the genome is graphically represented. They carried out this procedure separately for the highest and lowest bar sequences. They combine the two graphs for highest and lowest bar by subtracting the S&P index value of the latter transformer to generate the graph of the S&P index. In this graph, the S&P index equals 1 if the back DNA comprises only parent B genome. The S&P index equals -1 if it is of parent A genome only. And the S&P index equals 0 if both parents have the same S&P index as the genomic regions. As in most genomics intervals, the S&P index was close to 0.5. Correspondingly, the S&P index was close to 0. Such genomic regions were unlinked with a trait of interest. In contrast, regions containing cosinosa for trait difference between two pools yielded S&P index plots that appeared as major images like this. As was mentioned before, we investigated chilling-tolerant phenotypes of 331 with imbalance. Among which, the very chilling-tolerant D9046 and the very sensitive JN6 were selected as parental samples to construct the population. The reason why D9014, D9046, and JN6 were chosen as parental samples is that D9046 exhibits competitive architecture and resistant to disease, low temperature, and water logging, and it is widely used in maize breeding. JN6 is used in hybrid maize breeding due to its high grain yield. Both D9046 and JN6 are 80 level inbred lines. Among the active population consisting of 1,569 lines, we split out 50 extremely cold-tolerant lines to form an R pool. That is a resistant pool. 50 extremely cold-sensitive lines were screened out to form the S pool. That is the susceptible pool. We conducted genomic resequencing on D9046, DN6, R and S pool. The coverage depth of resequencing reached more than 50 times. We found highly contrasting patterns of S&P index graphs for R and S pool in these two regions on chromosome 5. Combining the information from two graphs for up and output, we made a graph of dirticy index, whereby the dirticy index equals S&P index of output minus S&P index of output. This reveals that most of the genomic regions show dirt S&P index equals 0, but some genomic regions exhibit positive and negative values of dirt S&P index. This may correspond to QTL, governing the difference between the R and S pattern. We calculated statistical confidence intervals of dirt S&P index for all the S&P positions with given real depths under the null hypothesis of no QTLs, and plotted along with dirt S&P index. The chance that dirt S&P index become higher than 0.35 as observed for the cornerstone region between 40.17 and 43.47 and between 78.50 and 80.29 mega. Is p-value less than 0.01 under the null hypothesis. Within the candidate two regions of approximately 5 matrices, 70 genes were included. We conducted GO and KEGG analysis on these genes. The enrichment analysis of GO and patient revealed their involvement in various biological processes, including responding to stimulus, cellular process, and metabolic process at all. Several coconut GO terms included memory part, and so on, and only two molecular function GO terms, catalytic activity and binding, were enriched. KEGG pathway enrichment exhibits that metabolic pathways and biosynthesis of secondary metabolites were the most enriched pathways. The results suggested that accelerated protein synthesis and division contributed to low temperature stress activation. Among the 17 candidate genes, only one gene 228 was reported in more than 8 tolerance. The remaining 69 genes had no experimentally validated functions. We therefore deduced putative candidate genes from severe homologous gene annotation. Among those, 26 genes may be involved in cold stress, and other 23 may be involved in response to other evacuatory stresses, such as heat, cold, ISOP, and abscisic acid disposal. Within the 70 candidate genes, the first of 2,515 affinities and alleles were identified as different between two printomides based on the BSC data. To verify the affinities and alleles and combine genes with allelic polymorphism bells lead to chilling-tolerance. Classical markers were developed and assessed in another 20 chilling-tolerant and 20 chilling-sensitive lines. After filling out synonymous or intranuclear variations, 921 affinities and alleles were identified by classical seeds. The results showed allelic polymorphism bells among chilling-tolerant or sensitive embryons in only 20 of those affinities and alleles, consistent with the BSC data. The other affinities and alleles were randomly distributed among the chilling-tolerant and sensitive individuals. We identified 20 SNPs within 6 genes. They are 3M40, 1D014, 321, 324, 325, 211, 212, and 223. Activities were not assessed at specific points in the candidate genes among the selected chilling-tolerant and sensitive lines. For example, at nucleotide position -1003 from A of the starting codon in strain 324. 12 chilling-tolerant lines contain a gene at this position, and the remaining 8 had a A. However, 13 chilling center lines had a A, 7 lines contained a G. Therefore, chilling center lines tended to have a A at this position, whereas chilling-tolerant lines tended to have a G. This base thus identified with Caspar for consistent biophysical results. So, a linear produced of a biometric cheese may be related to natural variation in cold tolerance. Next, we use RT-qPCR to compare expression levels of these genes before and after low temperature treatment of microtypes in the two parental lines, N046 and JN6. We found these genes show different expression pattern between the parental lines. They are prioritized for code-related functional study. This work was mainly accomplished by Dr. Min Yan and Fang Li. Thanks for Professor Qingpeng Sun from Beijing University of Agriculture for supplemental support for this work. Thanks for Professor Zhuran Zhao from Beijing Academy of Agriculture and Forest Sciences for his financial support for this project. Thanks for Beijing Academy of Agriculture and Forest Sciences for the project funding provided to this research work. My report ends here. Thanks for your attention. Okay, I finished my talk.
Thank you, Doctor. Now the session is open for queries. Any questions to Dr. Yunma? Thank you, Doctor. Now we'll move to the next presentation. Next presentation will be given by Dr. Apekshita Singh from Amity University, India, and the title of presentation is the Genetic Divergence Assessment and Selection of Core Genotypes in Indian Pigmented Rice. Over to you, Doctor.